Okay, can you guys see the annotation bar that I'm moving around, or just the laser pointer?
I see a laser.
Okay, you don't see a bar right up here? Okay, cool. Thanks for the input. I was just curious. Okay, so most RNA that goes through RT-PCR will have this poly A end to it, kind of like a poly A tail. It actually is very much like a poly A tail. So there will be this primer of DNA that will attach to that and start as a place for the reverse transcriptase to sit down. So what you can do for this is either have a global primer that will just start at all poly A tails and this primer will attach. Or you can have a very specific primer that will be for your own specific RNA of choice. So and I'm going to get back to this as well, but the CDC sent out a small test kit, and it shows basically like 4 primer tubes. One set of primers is specifically for the reverse transcriptase part. So that will bind to your specific DNA or RNA, sorry, the RNA of the COVID virus. That only will be amplified, and then you'll have less background that you're having to worry about. Then you're going to have a reverse transcriptase start putting in each of the nucleotides in bulk here. But remember that the T's are going to be replacing the U's here. Uracil is going away because this is not RNA anymore. This is DNA. And we call this cDNA because it's a complement to what the RNA was. And so it's complement DNA. We actually use this to make RNA libraries where we're seeing what an entire cell is transcribing and wanting to translate at one time. So we can look at what is being made. But in this case, we are just looking for that one specific virus genome. So we have this long, usually it's about a 30-minute window where we're having people, or a 30-minute window we're having reverse transcriptase amplify all the genomes as much as possible of the COVID virus. And then we heat and activate the reverse transcriptase. It is usually not as hardy as the DNA polymerase, which is for a higher temperature. Okay, we're not looking for detection here. We're just making the template that we'll then amplify and look for, okay? So in a thermocycler reaction, the heating reaction that we usually do for this, we'll have sometimes 30 minutes. In a fast reaction, we can have 5 minutes or even 1 minute of amplification of that DNA from the RNA. Does anybody have any questions so far? I've used a lot of terminology. Some of you have had polymerase chain reaction before, and I'm going to go into it, but I've used thermocycler, I've used primers. Are there any questions there before I go on? Okay, I'm having shaking of heads. Nobody's fallen into a coma so far. Day 1 is a success. So, there, that's what it was. Okay, so in the end, you'll have complement, and then DNA polymerase will fill in the opposite side of this. So you'll have double-stranded DNA. A's and T's, G's and C's, because it's more stable that way. Okay. Then we get into polymerase chain reaction. And basically, you put in what we call DNTPs, which are the deoxyribonucleic acids, the building blocks for the DNA. You're going to be putting in primers, DNA primers, not RNA primers. So you have to have 2 sets of primers, an RNA primer and a DNA primer. And this will be very specific for the area of the genome you want to amplify. In this case, it's a certain section of the virus. Okay, not the whole viral genome, just one section of it that is specific for not only coronaviruses, because there's a number of circulating pneumonia-causing coronaviruses that show up once in a while, actually seasonally, like coronavirus betas. Those are common in the hospitals, but they don't spread like what we're seeing with this coronavirus. And so this will be specific not only for coronaviruses, but for the COVID-19, which is why we needed to get that from the CDC. Now, one of the problems with the CDC ones is they didn't bring it out very quickly, the test kits, and they don't have enough of this primer being made. Don't know why. It's one of many ways that the CDC organization and the governmental organization drop the ball early and often. Apparently, there are more cases coming out, but or more test kits coming out, but I don't know. I haven't seen any evidence of it yet so far, so we're going with it. So you have these primers. You have the DNA, and it's double-stranded. So you have a 5' end, a 3' end, a 3' end, and a 5' end. And you're going to heat it up. And that's that denaturation step. And it's usually, they say 95 degrees. You know what? DNA, unless it's really, really, really full of guanines and cytosines, is going to melt at 90 degrees. So above 90, we'll just say that. It will denature, which means the bonding, the hydrogen bonds will break. It doesn't damage the DNA. It'll just go into the single-stranded forms. Okay, so we have a DNA primer that will fit on one end. It will bind specifically to all the nucleotides at one end of the sequence. And then we'll have a primer that will bind to the other side of the sequence, but on the opposite strand. So you have to design your primers correctly. Personally, I hate designing primers, but it is a key component of understanding molecular biology, so I've gotten really good at it. It's mostly the stress of if you mess up, you have to redo it. I don't know. It's I don't know, anxiety, whatever. Still, I got to do it. So you have the primers anneal, and this is the second bit where you're going to have the annealing, and 68 degrees is really high. You're usually not going to have things anneal at that point because the bonding is going to still be occurring while you're having amplification, and you could have errors or no amplification whatsoever. So when you're doing the annealing, this is just a slide that somebody used, I would or made, and I had it here. I usually say about between 45 to 65 degrees centigrade tops is usually your annealing temperature. And this is basically coming down off the 90 degrees. Everything's in this stew or soup. And then as it gets cooler, they're getting slow enough to actually start binding with a complement. So the DNA is binding by the complement here. So it binds, it anneals. Now you have that start of the railroad track for the DNA polymerase to sit down. And the DNA polymerase is sitting in this milieu too. It's that huge complex that sits down on this track. It doesn't need a promoter. It just knows where it needs to go. It's the wheels are running and you just need to put it down on the track. So as soon as it lays down on this, it'll start making complement DNA in the 5' to 3' direction, as shown with the arrows. The other strand, the complement strand, it'll start making it in the 5' to 3', but it is shown here as backwards because, well, we have to show what strand it's being made off of. It's then going to take up this primer and replace it with nucleotides. So the primer is going to be free to bind to another substrate later on. Okay, so the annealing is part 2, and that's about max 65 degrees. I can explain that if there's any questions about that. But then we have the elongation at 68 to 72 degrees, which is here. Okay, so far, any questions? Okay. Then this is where it gets kind of fun, because you go through multiple cycles of this, and you're going to have a more and more specific product. Now, this first time through, there's nothing to really stop this DNA from continuing to be made. Your piece of DNA that you're looking for, unless you have a specific end for it, it's going to keep making a copy of it until it runs out of time. Now, if you're doing an RT-PCR where you already have 2 ends that you've amplified, it's going to be a short segment instead of just a huge amount of DNA being made. So it's a little bit different in here with that one. So let's just say that the RNA that was made, the DNA segment that was made from the RNA has specific ends. It's, yes, Toby.
So I have a question in general. So how do you account for mutations during the elongation process? Like, do you add excision repair proteins as well, or what exactly do you do?
In the polymerase?
Yeah, in the PCR reaction.
Well, that is about 10 to the 12th. It will happen, an error will happen every 10 to the 12th time. So it's very, very precise.
Very negligible.
Yeah. If we were using like E. coli, that drops it, the error increases by about 1,000-fold because it doesn't have that self-editing ability. So basically, as the polymerase is making the, putting on the nucleotides this way, it's double checking the ones behind it. It's like double checking the track behind it.
Oh, okay.
So we have less problems with the error-prone thing. The reverse transcriptase sometimes has a lot of error to it. It's part of its design to make errors. So it's much less good at doing that. So the less you need to use it for long segments, the better. So yeah. Did I answer your question, Toby?
Yeah, that helped.
Okay. Alyssa.
Just to clarify, the new DNA strand that's being made is moving 5' to 3',
Yes.
but technically it's moving backwards on the original.
Yes. Yeah.
Okay.
On this one, right? That you're talking about?
Yeah.
Yeah. Because this one was the complement of this one.
Okay.
Yeah. So when you split it apart, we're still going to, we have a primer here to amplify the section. It's just going to be the opposite strand.
Okay.
Yeah. No, it's a good question because you have to kind of think about it. We could just go amplify off of this strand, but we would not amplify everything logarithmically or exponentially like we want to if we didn't have it. Okay, good questions. Okay, then we take both of these segments and run it through the whole cycle again. We denature it, so these two sets of DNA that are complementary base paired together, we melt those hydrogen bonds and split them apart like we have in one. Then the primers sit down in annealing and anneal to those sections, and then we have elongation again. Except some of the segments in this case will not go forever. They will go just as far as the beginning of this side. Because we're going to have more and more segments that only start at this 5' or only start at this 5' that the primer is binding to. So we're going to get a very specific section here. So some of it will bind to the original segment and make tons of sequence that way. Some of it will bind to this already made segment that has a distinct end to it. And it can only amplify up to that point because it doesn't have any more nucleotides to make a complement to. So you're going to get more and more of these as we go on. Proportionally less of the original DNA shown in green, and we're going to have more and more of these smaller segments of the DNA being made. So you're going to have a very specific size being amplified again and again and again and again. Yes? No? I'm getting, oh, I love this. I'm getting more response than sometimes in class. I'm joking. Your class is great. Okay. So we have this amplification, and this cycle can go on some 30 cycles. I love that doggy, Beth. I will have kids sit on my lap sometimes, so just get prepared for that. They're pretty good, and they might learn something too about PCR. But you'll have this cycle go multiple times, sometimes 30 or 35 cycles. But in a reverse transcriptase PCR, you have to have kind of a cutoff on how many cycles you can do before you go, yeah, nothing's there. It's a full negative. So usually what they'll do in RT-PCR or even PCR for testing on disease states or especially infectious diseases is say, okay, if we don't get any amplification or positives by 20 cycles, it's not a positive. So they have to have a cutoff time in these cycles. Okay. Now, what happens next is we have to detect it, and we are in the stage of detection. These RT-PCRs that they are using for the coronavirus are real time. So it's a RT in case of this. It's a real time reverse transcriptase. And what the real time means is that we're detecting it as there's amplification of DNA. We don't have to wait until there is a gel to run it on. Okay, sorry. There's a lot of people talking to me. Now, what we will do here is put in something called cyber green. It sounds really cool. Roche developed it, but it's used by many people now. And cyber green will only, it's a fluorescent molecule that will fluoresce specifically when you bind to double-stranded DNA. So you can have it in the detection tube, and most of the time you will not see it at all. You have to have not only a fast cycling of these this amplification. We'll talk about how fast it is in a little bit, but you also have to read how much DNA is at every given cycle. So when we're talking about this amplification, we will go through one whole cycle of denaturation, annealing, and elongation. And before we go back to denaturation, on real-time PCR, we will have a fluorescence reader read how much fluorescence is in each tube and give you a number for that. Then it will go back to denaturation. The, oops, doggone it. The dye that was bound will fall off again because the bonds have melted and we don't have double-stranded DNA anymore. So it's a real-time, that instant, how much DNA is there, and then it falls apart again, which is pretty cool. Now, what you will see here, this is a actual reading off of a thermocycler. You're looking at fluorescence, normalized fluorescence for what is a negative versus a positive, and we can talk about that, but it kind of depends on the test. and then how many cycles there are on the bottom. So you go to 5, 10, up to 45, which is highly unrealistic. It usually only goes up to 35. But you'll see this in individual positives coming up where they can detect DNA fluorescence based by the cybergreen binding to the double-stranded DNA, this amplification happening already at 10 cycles. We couldn't do that by eye or even by a gel doc, a really nice gel doc that we have at school, which is going to sit there gathering dust. But you can see it happening in real time. And so you're going to start seeing positives like this come up at different times. And you see a bunch of them show up at about 20 to 30 or 25 cycles. Usually at 30, you're going, yeah, it's not going to, there's no positive here. But you're starting to see once it gets past this threshold, which is usually what we consider the background fluorescence. We won't count anything positive below this because it might just be fluctuations in fluorescence from primers binding together or something like that. So this is what we'll get as a response then. The light cycler I worked on was one of the early ones, so it could only do like 25 at a time. So you could name each one of them a positive and negative control and then like 23 other samples, which was nice, but it's really slow. But you would be able to say, oh, number 5 came up positive really early. That's a strong RNA signal for West Nile virus. Great. Does anybody have any questions about the detection? It's a really powerful tool for detecting this in real time, so you don't have to wait overnight or wait for a gel to run or not detect some of them because you can't actually see it. Okay, well, if you have questions, I can do this later on. Now, what they do now is have specialized tubes and a specialized machine. This is a Roche light cycler. One of the early ones basically looked like, I don't know, a big tube with a smaller tube on top. It was kind of ridiculous, and it could only do 25. The whole idea of these fast light cyclers is you can amplify DNA in not hours, you can do it in seconds. So you can do an entire cycle of denaturation, elongation, or denaturation, annealing, and elongation in less than a minute. So that means you can amplify everything and detect everything in such a small amount that you'll be able to go through this very quickly. So each tube has all the DNAs, the DNTPs, it has the primers for reverse transcriptase, you have the reverse transcriptase itself, you have the primers for DNA amplification and the DNA polymerase itself. And you have a buffer and things like that, and of course the sample itself. Then they have these tubes that are glass. Glass transfers, a very thin glass transfers heat a lot faster than plastic will. So they have these specialized glass tubes, and then they have this really fast, I gotta work on this, a really fast changing heat sink. It's all by hot air. It's basically a convection oven if somebody bakes around here, which I do. I'm gonna make buttermilk rolls later on, and maybe some bread, and maybe some sweet rolls for the kids, because sugaring them up while we're trapped inside is always a good idea. Basically, you can have the temperature change in these tubes very rapidly. So rapidly that you can denature in 5 seconds versus something longer. So you can put them into these trays on the light cycler, and it all slides in. This is beautiful compared to what I had. And then you can see the results in real time on this readout, which you can see here. And you'll basically see these curves show up They're not as usually beautiful as that. They're kind of like this, but even messier. And you'll be able to see, okay, once we get to this line, that's a positive, and you can see it versus the cycle. So that's a real-time RT-PCR that they're doing. Some of the things you need is the correct sampling swabs. You have to have the ability to do the sampling swabs. So the sampling and the equipment kind of go hand in hand. You have to have coordination with state, local, and federal authorities. You have to be able to know who to test. And that has been one of the first and big issues with this COVID response. They had a very stringent response that they didn't tell people to stop doing for a long time. They were only looking for people who traveled overseas or were on a cruise ship. Some places are still only testing those. That meant that if you had all the symptoms, fever, a dry cough, a pneumonia-like state, and it wasn't influenza and it wasn't respiratory syncytial virus, so RSV, they still could not test you for COVID because you did not meet the stringent guidelines. Well, did you travel overseas? No, I didn't. They didn't think there was person-to-person personal spread in the communities yet for the last 6 weeks. There's evidence, pretty good evidence, that it was spreading person-to-person in Washington for that amount of time, which is why their numbers exploded overnight. Once they started testing, they found out that a lot of people had it. I've been talking with people in the hospitals, some of our students who are doing scribing, as well as some of the doctors. And Brookfield Hospital, they've been seeing a lot of non-influenza or negative for influenza pneumonias and respiratory issues at the hospitals for the last 2 weeks, and they have not been allowed to test for COVID. Not only that, Brookfield doesn't have the test. Only Froedtert and St. Joe's has the test, as well as the state health lab. So you can't even do the test if you don't want to, or even if you want to. So there is a acronym in the military called FUBAR. That is what the situation is now. Because there wasn't coordination, because there wasn't information or just wanting to find out if it was in the community, they didn't allow people to test for it. Now that they're allowing people to test for it, all of a sudden our numbers are going through the roof. Well, it isn't because all of that isn't because of the spread now. It was because of the spread 2 weeks ago. Like I told everybody, not that I'm going to rub it in too much, but you guys believe me, and I appreciate that. Anna.
Yeah, this is weird. I don't know if I'm supposed to raise my hand or not, but I did. So what does FUBAR stand for? One, that's my first question.
I shouldn't have said that.
Because you named our syllabus that, so now I feel like I need to know.
I didn't mean to keep that name that way. I'm going to rename that. It is
Just say it.
It's messed up beyond all recognition. Or so
I'm gonna Google it.
Yeah, no, it's fracked up beyond all recognition. So basically, the frack is a Battlestar Galactica swear word. There. I'm such a nerd. I could do a swear word in Farscape. So they had to make swear words for Australians, otherwise they couldn't act well. So they had to make this a whole fake swear words for space Australian sci-fi. So I know that one too. But yeah, fill in the F word that the military usually uses. Sorry, I was a military science. Gonna go into the military, so it's my automatic. I will change that on the syllabus now. Thank you. Sorry about that.
It's all good.
Yeah,
My
that's a
second
Christian
question.
demonstration, right?
I have another question, and it's
Sure.
just about, like, I've been hearing a lot. See, I don't know if this is true or not, but like
Sure.
you could have COVID, but your symptoms are a lot less severe than what they're talking about,
Yeah,
and then you still like possibly have it and are spreading it. Like, could that be like that's a possibility? That's a
that's an
thing.
absolute 100% true.
Okay.
You can even have, it turns out they were doing a study on this. I just saw it yesterday evening, that about a third of people don't even know they have it. They will be asymptomatic completely and spread it like crazy. Then there are going to be about 80% who get infected, roughly. They were saying 80%, it might be 70% that get infected and have symptoms. A lot of these people, the people that have symptoms, a lot of them are going to have very few, a mild case. In a mild case is you're going to have a cough or a fever and recover fairly quickly. Then you're going to have some that are moderate case that have a cough, fever, and the cough gets to the point where they're coughing up crap. And then you're going to get 12 to 15, even some people have been saying 20%. It depends on the population. It's weird. That will have serious complications where they're having trouble oxygenating the blood. Those people will have to get some kind of hospital time. Sometimes it's just getting some drugs that'll help them breathe better. Sometimes it's oxygen. Sometimes it's a ventilator because their lungs are so full of fluid they cannot breathe. Yeah, it's the real question is the amount of people, because we don't have that percentage of people that are going into the hospitals for influenza in the vast amount of people. So we can treat them if we have enough people to do it and enough equipment to do it. But right now, the numbers are going to start doubling. We're almost at the doubling every day stage. We're doubling. It's not really doubling every 2 days like they're saying, just because we're getting more positives. We're going to have people who are getting symptoms that have to go into the hospital doubling in about 3 to 5 days at this point, but it's still not good. We're going to have double the amount in the hospitals in New York, probably in 4 days less. Bless you, Silas. And it's like the Brady Bunch. Yeah. And I never watched the Brady Bunch. I'm not that old. But yeah, we're gonna run out of ventilators really quickly. They have run out of ventilators in Italy already, and they've made the choice, at least regionally in certain areas, to say we are not going to treat 60 years old or older. Yeah, we thought 2019 was bad. Having the thought of having to choose is unthinkable outside of wartime. Hopefully we stopped it. Oh, yes, Emily.
So is that kind of why there's a lot of people in Italy who have died?
Yeah.
Or is there other factors too?
There's other factors. They have a slightly older population, a larger older population. They also have closer quarters, and we think also culturally they have a lot more close contact with how they greet people and things like that. They're apparently friendlier than many of us, but I'm not going to comment on the U.S. Possibly that's why Germany hasn't gotten as many people really sick, but I am a German, so I mean, if you've been in the wells for very long, it's you can know somebody at church for 15 years and then give them a casserole as a sign of friendship, And that's about as close contact as we get sometimes. It's just the way we are. I'm trying to break that, but I'm not going to be hugging anybody anytime soon. Not only Title IX, but just COVID-19. There. Yeah. So they have an older population. They have a lot more people that smoke. They have an excellent healthcare system, but they have a whole healthcare system that's based off of reactive care. And they don't have as many doctors that are there on the spot. That being said, they're overwhelmed. They got caught with their pants down because they didn't expect to have an outbreak go from China to them. Mm-hmm. And it's a horrible situation. And they had another surge yesterday of cases and deaths. It's really bad. They do not have enough ventilators for all the people that need to go on it. And I would like to point out that the median age of people put in critical care is 65. That means it isn't everybody 65 and older. It's half of them are above 65. The ones below 65 have a better survival chance. We're also getting people who are 20 years old being put in intensive care and on ventilators. They're just more likely to survive. So all the people doing the stupid spring break on the beach and everything, yeah. It's like, yes, Connor. Yeah. There you go.
Sorry about that. Yeah, so I heard that people who are young have a chance of developing like a chronic lung condition after they are over with the main symptoms of COVID. Is that a lie? Is that kind of true? What is that?
It's hard to say. This is a good question. All these are great questions. It's hard to say. I wish I had a better answer. This is a developing situation. It does seem that a small percentage of people who recover have this scarring on the lungs, almost like COPD or emphysema or something like that, which lowers their ability to exchange gases, of course. We do not know if it's going to be a chronic thing. We do not know how prevalent it is. It was one study from China. And there's a lot of papers coming out from China right now. And it's really, I have the hardest time understanding how we can accept what's coming out, because they were lying about the original outbreak. So they're using those incorrect numbers to begin with. Now, it might just be that they're using a log scale and just taking off a 0, which is what we think of all the numbers, in which case the numbers are still somewhat relevant. But that was a small study. We don't know if it's long-term, and we don't know how many people are really going to get that. I mean, that's scary in itself, that some people would be damaged for life, and they're young and are not going to be able to make it up a set of stairs without a cannula and some oxygen tank. I think we all still need to remember it's a percentage, a very small percentage that we're seeing right now, and we don't have a really good study on it. So we're just going to have to worry about that bridge burning at the time we can take care of it, and wait for the data. That's really what we need to do. I don't think most young people, unless they have some kind of underlying lung illness, are going to have that problem. That's my kind of educated guess at this point. Maybe. It's a good question. I just wish I had a better answer. Ouch. I can't laugh too much. Yes, Anna.
I know that we don't really have enough like test kits, and so we really don't know how many people are affected, but I know that you are probably thinking about it. Do you have like an estimated guess of how many cases are in the States like right now, or like you're saying that it's going to double every day? So are you like...
I don't think it's going to double every day. I think now that we're testing more, it looks like it's doubling every 2 days, but I don't think it is, just because we're looking now. That being said, if we say there's 35,000 cases as of right now, I have the COVID, actually I can, let's see if I can bring it up. Where did you guys go? Crap. There. Nope, that's just you. I have the database up. Italy is still going strong. We now have 42,000 cases, 507 deaths. That went up in the hour. And we also have 450 that are serious. So those are definitely on ventilators. A lot of those are in New York. So they're running out of ventilators quickly. I would say if we have 42,000 cases, we probably would have about 80,000 cases at the very least. In Wisconsin itself, right now, they're saying it's laughable because they're saying we have 380 cases. I'm saying we probably have over 1,000 because we're still not testing. We don't have enough tests. We don't have enough test kits, and we're frankly not trying hard enough. Bree.
Isn't there something though where like they won't test you unless you have like certain amount of criteria, like unless you've traveled to whatever country or like you know that you've been in contact? Like,
yeah, and
I mean the odds that you know that you've been in contact with somebody is like, I don't know.
Yeah. They keep testing people who traveled overseas. I think they should stop that because we know we are in the thick of it. We know there's person-to-person transfer in the US, period. We know this. I don't know why they can't get away from that, frankly. It's wasting time, and the whole idea of the test is finding out where the worst cases are, you know? So why aren't they doing that? That comes back to the coordination, logistics, and willingness to test what they need to. We're not testing enough of the right populations. And on top of that, we don't have enough of the test kit. So we don't have enough of the primers. On top of that, we need to prep the throat swabs. We need to do an RNA extraction off of those. And we don't have enough of the kits right now. Those are usually coming from QIAGEN or another DNA and RNA preparation company. Though we usually do QIAGEN only because those are very, very well vetted kits. They're easy to work with at high throughput. They are the Cadillac of, or whatever expensive car of the molecular biology field. They can't keep up with the entire world wanting their product at once. So they're trying to, but we don't even have the prep kits for that. On top of that, we don't have the RNA polymerase RT-PCR master mix with the DNTPs, the RNA reverse transcriptase. We don't have it with the polymerase. We don't have this mixture that is guaranteed to work in enough amounts. I have the RT-PCR master mix at work. I don't have the primers or the RNA prep kit. Connor, did you have your hand up?
Yeah, going back to like the amount of actual cases in Wisconsin, my mom works for Aurora and she oversees a lot of the hospital patients.
Yeah.
And she says there are a lot of patients who are dying of atypical pneumonia that were pending for a COVID test, and they died before the results could even come back.
Oh,
so
right.
She's thinking that the numbers are not very accurate because in a lot of cases they can't even get the tests, and you know, some people may not even be tested and they just die of atypical pneumonia.
Yeah.
So that's just another awful thing that's going on.
Thank you for sharing that. It's very distressing to show how correct we are about this. But yeah, the sampling, there are places where people are dying and don't have the sample, aren't sampled. And the originally 2 weeks ago, one of my cousins is in charge of pharmacies in and around the Twin Cities, and he got word that they had a positive case on a Friday 3 weeks ago in Mayo, at Mayo Clinic, the Mayo Clinic. Okay, and then the CDC said, wait, you can't announce that. We have to double check it at the CDC ourselves in Atlanta, Georgia. So they said it was going to be 4 days. They were allowed to announce it the next Friday because they finally got the test back. So Mayo has developed its own positive or own in-house kit. There's a number of places just saying, okay, CDC, you're a joke. We're going to make our own kit. And they're using that as reason to detect. Yeah. Beth.
What do you know? Do you know if there's like false positives or false negatives? Because I know there was at least, I can't share too much because I probably shouldn't even know this, but my mom's working from home sometimes, so she can't avoid those things.
Right.
There was a case where someone tested negative and then they got tested again and they were positive. So do you know if that's a thing that's happening or?
It's actually, I'm not surprised on that one because you can be not shedding virus at the time of your detection, and then later on you could be shedding virus, and then you could be a positive. There is a possibility that let's see, let me think about this. If you have a small sample size, you can have a higher error with false negatives. But as you get into a much higher amount of people being tested, you can have false positives too. Just because it's every test, and we're getting into epidemiology and test information, there's always an accuracy and a precision amount to the test. And then that will give you the amount of false positives, how many times it's going to be absolutely correct, and how many times it's going to give you a false negative. And we want a test that is going to be 97, 98% positive, as in it will always give you a positive when it's supposed to give you a positive. But on the reciprocal side, it's difficult to find a test that will have 97 or 98% negative, as in it will give you a negative when it's supposed to give you a negative. As you get higher in the numbers that are testing, you're going to find those more often. We have that with ELISA kits and immunology kits, which hopefully we'll get into because then we don't have to worry about sending it to the health lab. The UK has developed a kit that's like that. So it's fast, easy for people to do. It's going to be like a strep test. These false negatives and false positives are going to happen. I don't think it's bad that they're treating it as a positive and reacting as a positive. If they're just going to isolation, sure, why not? No harm, no foul, really, at that point, because we all should be. Did I answer your question, Beth? Ashley had her hand up? No? Okay, sorry. Brianna.
Somebody is asymptomatic, like say they do get tested even though they're asymptomatic, would they still have the possibility of testing positive if they're shedding virus?
Yeah, they would.
Okay.
Because they're still actively infectious.
Mm-hmm.
So, yeah. This is the hardest thing for people to put their heads around besides exponential growth, is that it could be floating around and they can't see, taste, or smell it. It's like training people to work with radiation. You can't smell it, you can't taste it, you usually can't see it. You have to act like it's there all the time, which makes a lot of people paranoid and they can't deal with that. It's just training or lots of paranoia, but it isn't paranoia if it's actually happening. So I'm not paranoid. Okay, so we talked about, are you seeing my PowerPoint? Yes, no? Okay. So we basically talked about the reagents and prep. Some of these just take time, and as we get overwhelmed with samples, it's going to take longer time, especially if we're running low on the sample kits. The equipment is expensive and very specific. You can't run this just on a regular thermocycler like we have. You have to be trained specifically in RT-PCR. You can't just throw this together. You have to get trained on these machines because they are not like other machines. You have to know what the limit of detection is. You have to know what a false positive is going to be. You got to know what kind of errors can creep in and how to troubleshoot on the machine, because there's always going to be mistakes. It's a fairly rigorous training. It's supposed to be idiot proof. It's not, because I'm on it. Yeah, it's a highly technical test. It's very sensitive, but it's highly technical. So, a good false positive for you. A couple dogs have tested positive. This does not mean that they actually get COVID. However, a number of SARS species have been shown to infect or be spread by dogs, cats, camels, bats, not armadillos, this weird armored thing that looks, I cannot pronounce that. Well, something that people eat, apparently. So it can be spread, but I don't know why they were swabbing a dog to start with. I honestly don't. But there is no indication that it can be spread by canines or dogs, or canines or cats, so it's probably okay. Yeah, there's a lot of problems, and honestly, Fauci has been setting up this whole system to react against the next outbreak for the last 10 years, even longer. We have been trying to set up factories that would make antibodies and vaccines for anything at the drop of a hat. They can be adapted to make vaccines quickly. But we haven't been doing the basic research or setting up and putting enough money into making sure we have a detection network. We had one in the Bush administration just because of there was a lot of money thrown at biodefense after the anthrax mailings, because we thought there was going to be a lot more of them. So we had hospitals set up BSL-4s in every region. We had BSL-3 networks that would filter into that. That was Marshfield Clinic. I was setting that up and I was part of it. And then we had a lot of basic research for detection methods of all these things. We pumped a lot of money into it. And then the money started going away because they didn't see instant results, or that it was really helping anything because we didn't have any more bioterror attacks. The public forgets very easily. Politicians forget even faster. That's all I'm going to say about that. But I would anticipate that the research money for those that survive is going to be quite good for the next decade or so. We might even have some researchers die off at the top, making room for others younger. Not that I'm hoping that, but it's a real possibility. It's funny how many microbiologists do not take care of themselves, speaking from experience. Okay, now, what I'm going to do for the rest of the presentations is we're going to have a brief meeting like this. I'm going to have you look at a brief talk about a technique before this. I am going to have an assignment of techniques unless people want to give me an idea of who wants it right now on each technique that you're going to write about. You're going to be given we are going to be talking about a possible scenario on a piece of, or a technique that we're going to use. Like I'm going to talk about ELISAs. We've talked about RT-PCR. In the next couple of times, we're going to talk about some phage identification. And then we're going to say, okay, say you need to identify phages in a water source. How would you go about this? And we're going to work through it and get the ideas for controls, how we would test it as a group. So it's going to be a group think tank about how to do an experiment. And those people who are assigned to that one are going to be frantically writing this down, because they're going to be writing up a paper that is based off of detecting, say, phages in water supplies, a specific phage against a bacterium. And then they're going to have to write up the materials and methods that we devise in class, just as if we were about to do it in class, because that's how I had planned to do it in class. Does it make sense so far? Yes, no? Okay, I'm getting 50% nods, 70%. I'm gonna go with it. So we have the technique. I've talked to you about the technique. We're gonna put out a scenario that we're gonna develop an experiment for all together. Then there is going to be a group that is going to write this up as a paper, as talked about in the syllabus. That part is sticking with us. And I'm going to give them data. This data is what they're going to analyze and determine if they actually have that phage, and if so, is it specific for that bacteria or whatever. And then they're gonna write up the results as if that data was real. So I'm just going to give you the data, and you're gonna respond to it in a real as if it was a real situation, and write it up as a paper. Then you're gonna have conclusions, discussion and conclusion section where you're tying in primary literature. And that is going to be your paper. Any questions so far? Okay, so let me pull up the, I'm gonna pull up the, yeah, I gotta really change that foobar thing, you're right. Holy cow, I apologize about that. That's how I'm thinking of it. Let's put it that way. Nope, that's 201. What in the world? Stop, 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 stop. I forgot about pulling this one up, so I apologize. Yeah, I gotta stop naming these things foobar. I may have sent that to the head of the department that way. Whoops. Okay. So I will, oops. I'm going to share this with you. Where is it now? There. Okay, people can see this? Okay. So we're talking about, and I was going to talk about development of this, but this has been beaten to death, and I don't think people can deal with it right now. We have one group of people who are going to do the dilution series experiment. So that's 3 people. That's Bree, that's Kara, and Alex, who I hope is not dead. Then we're going to go into the Ames test and plasmid detection. So that's going to be kind of two separate ones. We are then going to go into the flush test, which is basically detecting whether or not feces is flung into the air when you flush the toilet. We actually did this experiment last year. It was really fun. So we're going to do it again. Well, not really. You guys get to block off a whole bathroom with crime scene tape and then sit in there with lab coats and PPE. It's great. And flush the toilet a lot. I am going to talk about using or detecting a virulence factor, which is biofilms. So that's going to be one of them. And then ELISAs. I think I might move ELISAs up higher because that will take some time. So basically, I would look at the Ames test for mutagenesis, the plasmid detection, the flush test, and then let's finish off with the ELISA so you have enough time to write up your paper. So let me write these down. Oh, I don't have the phage up here. I cut that out. Ames test, plasmid detection, flush test, and ELISA. Okay. Does anybody want to have the Ames test, which is basically detecting mutagenesis or how carcinogenic a compound is? It's either that or tracking plasmids throughout a hospital with resistance markers. It has resistance genes on it, and it's going through a hospital, and you have to detect where this plasmid is.
So are we still writing the paper as partners, or is it individual?
Yes, you
now?
are. Yeah.
Okay.
Partners.
I mean, I would have taken the Ames test, but Anna is my partner, so it really depends on her.
Okay. She gives it a double thumbs up.
Okay, cool.
I can't.
Wait, sorry, what?
Okay. What about the plasmid detection?
Yeah, I'll take the plasma detection. Oh, unless someone else wants it. Did someone else just talk there?
Yeah, I said Tierney and I could do it, but if you want it, that's okay.
We can have multiple people doing it. It isn't that we're doing a presentation. We can do a paper. So, especially now that we're in the thick of it, yeah. Most of the rules are out the window, except we should all wear clothes when on Zoom. One of the teachers told me that we need to have that rule, and I wanted to ask him why. But Connor, who's your partner?
Matt is. So if he's okay with that.
Yeah, that's good.
And okay. Emily and Tierney, yes?
Mm-hmm.
Okay.
Yep.
Flush test. No feces? Okay. Well, we'll think about that. And the ELISA.
I would like to do the ELISA.
Oh, okay.
Ashley, are you cool with that? Cool. Ashley's my partner. We're going to do it.
Okay. Now, let's see. Gretchen and Bethany. I think you're only... Wait, Silas, who are you with?
With Katie.
Who's Katie?
She's not here.
Oh, okay. Yeah. So Silas and Gretchen and Bethany, you must make a choice.
I don't have a preference. Gretchen, do you have a preference?
I don't really care.
We can do the flush test since no one's done it.
Yay! Okay, thank you.
I'm good with that.
I have some actual real data for that, so I can give that to you. It'll be fun. But at least you have some background about aerosol. You can look up already aerosolization of bacteria and fecal contamination in bathrooms and things like that. So everybody can start doing their intro on what all the information is framing their question. And let's see. Silas, gotta talk to me.
Yeah.
What do you want? You want to
Whatever's
think about
left.
it for a
I
bit?
don't care. I'll
Um,
talk to Katie too and see what she wants.
okay. You can, I would say you could do an ELISA, a flush test, or an Ames test as well.
Okay.
I think that since Katie is not here, you need to take the
Well,
reins and make an executive decision.
We'll do the ELISA then.
Okay, there we go. Is Katie dead?
I hope not. She was in self-quarantine last
Yes,
I
she
heard.
was.
Yeah.
That might have been my fault. I may have, no, it shouldn't have been my fault, but I made sure to let the administration know that I was not in favor of them coming back to school after being in a highly infected area, even though they weren't testing at that time. But that's okay. That's all right. Hopefully she's not dead. She's probably okay. Okay.
I do have a question though.
Yes.
So when exactly will the paper be due then?
I say that if you can get it done early, the more the
Okay.
merrier.
Mm.
I'm going to try to, what I could do if you want to get it done earlier is kind of put some of these together in a longer lecture time so we can talk about both of them, and then we can get them done a week ahead of time, and then you'll have more time to talk about it, more time to do the paper.
Okay.
Yeah, this is going to be kind of messed up. I can talk about the Ames test and the plasma detection next week. And then I could do the flush test and the ELISA the next week. So everybody has, and then we have some class periods where if you have questions and just want to show me what you have, I can give you some commentary on it and give you some updates on it for the last 2 class periods, 3 class periods, and give you some help that way. Beth.
So did you say we are doing a presentation or no?
Did I say presentation? I meant paper. Sorry.
Okay. Well, you were
want to
saying
do a
something about presentations earlier, so I just got confused. It's just a paper?
Yeah, no, sorry. I'm seeing most of the people that are in my microbial pathogenesis class again. And that will have a presentation. Here,
Okay.
tests, quizzes, and then a paper in the lab.
Okay.
Yeah, and the lab written test, but that's going to be a lot less onerous since we're not in the lab anymore. So I'm going to test a lot on the things that we talked about and did in the lab at the first section that everybody took copious notes on. And so like, what does this media, is it selective or differential? If you saw that it was purple and shiny metallic, what would that mean? Some basic stuff like that. I'm going to try to keep it down to short answer, like a single word answer or short answer. But it's going to be, I usually have a skills test in there. That's not going to happen. I usually have you reading results. That's going to be greatly reduced. So it's going to be a fairly light test. I will give you a review sheet on that. And I might have you explain polymerase chain reaction, the basics of it, things like that. But it is going to be greatly reduced. Tierney.
Okay, so next to the plasma detection, it says there's a handout instead of pages. Do you have that handout so we can like start looking at it?
That is, no, I don't. I will get it to you though. Okay.
Okay.
There, you know what? There are some pages in the textbook or in the lab book. I will find it because it does talk about, there's an older method of tracking plasmids called a, PFG, pulse field gel electrophoresis, PFGE. It's basically wiggling the gel with charges to get the all the plasmids separating out. But we have a better way of doing it nowadays, which is just sequencing. And a lot of yours is going to be tracking it through the hospital. I have a paper in which they did the exact same thing for a real outbreak at the NIH. And one of my friends actually did the tracking of it. So what I'm going to do is give you that as an example of how to do it. If that is better.
Yeah, that's fine.
Okay. Just remind me, a gentle reminder if I don't get it to you by the end of the week. Okay. I keep me, there's a lot of things juggling. So, Alyssa.
Can we get the handout for the ELISA then as well?
Oh, absolutely. Yeah, I have that.
Cool.
So
Thanks.
I should update all those so you get the basic information, and then I'll give you the situation. Yeah, that's a really good point. So I'll get that up this week. Okay. So everybody has a fair starting point. That's a really good point. Thank you. Other questions? Yes, Bree.
Can everybody send their data so I don't have to keep harassing you via email?
Yes, please send the information about stuff. Beth.
So for the flush test, is all that going to be collected like online through articles and whatnot?
I have a protocol for that.
Okay.
That I will give to you, and then what I'm going to have is people do a think tank on how to do this properly. Okay, so if we think of something really good, I'll tell you to write it down.
Okay. So there's no handout that we'll be getting but there will be a protocol. Like it's not listed on the table.
Well,
but there
that's your
something.
handout. That's your handout about it. There are papers that I will want you to try to take a look at. I will try to find one of them. There isn't a lot of information. This one was an urban legend that I was doing for a Mythbusters kind of format. I like doing the Mythbusters because it ties it into real life stuff. And somebody thought that we need to hide our toothbrushes and not store them in your bathrooms. So we're deciding to test that.
All right. Thank you.
Yep. Mm-hmm. Questions? These are all good questions. You guys are handling this like champs. Nobody's screaming. Weeping is okay. Let's just keep it on mute for that. I'll just turn off my video feed if I start weeping. Or if there, just please bear with me because sometimes there might be screaming. My wife, my wonderful wife, has break this week, and so she's dealing with the kids while I'm doing this, but next week it's going to be Lord of the Flies here, man. I'm not sure I'm going to come out on top. Let's put it that way. So we're going to do this to the best of our ability, and we're going to be really flexible, and I'm going to be very understanding, and I beg you to be at least a little bit understanding of the stuff going over here. I'm going to try to get you as much communication and as much being understanding of everybody's situation that might change from week to week.
Just to be sure, you said we don't have 9 a.m. lecture till Friday, right?
Yeah, I'm gonna try to. What I'm gonna try to do is at least have that. I started out saying that, and I'm realizing that a lot of people still aren't going to be able to make that. And I've already recorded lectures for the next 2 weeks in case everything goes wrong. And I almost said FUBAR again, but but if everything goes wrong. So what I might do is come online at 9 a.m. for your own specialized office hours. In case there's any questions, and I'll be there for an hour. If nobody's there, I'll just be doing stuff on the computer. I'm pointing to my other screen. I'm not insane quite yet, but that's what I'll probably do is meet online at 9 a.m. and you'll have the lectures recorded. I think that's the fairest way to do it. I've been breaking it down into sections so it isn't too long, except for one bit so far. Know your virulence factors that are, I think, are important enough to talk about. Know your basic Latin names for some of these infections. And I'll give you a quiz to give you an idea what I think is important. I have a long time limit on those. I am okay with you using your notes because this is a completely messed up situation. Also, I can't stop you. So there's a lot of the faculty that are going, well, what if they cheat? And I'm going, you know what? They're learning something anyway. And you guys wouldn't be cheating because you guys are wonderful people. Anyway, either way, you're learning something, and realistically, how am I supposed to control that and get it bent out of shape about it? So we all have to relax and ride the waves into the calm placidness of summer and insanity on the other side of these rapids. So hang in there. You guys are doing great. Thank you for the questions. I miss you all. But I think some people are more relaxed just hanging out on their couches or whatever. So maybe that's the best learning environment. Maybe we'll learn that this is better. I have my doubts, but we'll see. Well, I mean, I think everybody's really going to appreciate the difference between online learning and face-to-face learning. So, but you guys are doing a great job of being interactive, and I really appreciate that. That helps me too. Unless there are more questions, we can get done for today. It's wonderful to see you all. So thank you. I'm going to sign off. I'm going to stop recording.